Hello everyone, and a warm welcome to today's Focus On session provided by GenQA. Today's session is on acquired cytogenomic nomenclature, presented by Dr. Ros Hastings. Ros Hastings is a consultant with GenQA and also the chair of the ISCN Standing Committee.
Good morning, good afternoon, depending which country you're in. And we're going to talk today about the acquired cytogenetic nomenclature. We're going to talk about the basics. So we are going to talk about ISCN, what it actually is, talk about the basic principles, the building blocks. Some of this will be repeating what we did in constitutional, but there are differences for hematology and acquired disorders. Then we'll talk specifically about acquired ISCN, the FISH ISCN, RSI ISCN, Array ISCN, and the online version and forum. The ISCN committee is a standing committee. It's done through elections from the genetics community. There are currently 8 members, and I am currently the chair, but Sarah Moore from Australia is the deputy chair. There's now an online version of ISCN for everybody, and you can access this online version if you have purchased a hard copy for 12 months once you've logged in. We are now using and marking all our EQA submissions against ISCN 2020. So just to give you a bit of background, this is the ISCN Standing Committee. There's obviously the HGVS and the Human Gene Nomenclature Committee, and there may be in future an HGVS Pathogenicity Committee. These all now will report to the Hugo Standards Committee, which is just being set up, which then reports to the Hugo board. So there is a bit more governance in the whole way that we are working. So just go through some of the basics. We talk about metaphases. This obviously is a constitutional case. You'd like to see this in acquired, a karyogram, and the ideogram. And that's just so you understand when I'm talking about the nomenclature. The karyotype is the use of nomenclature to describe normal and abnormal constitutional acquired cells complement of an individual or normal tissue cell line. So chromosomes and ideograms. So when we talk about chromosomes, they're only visible with 2 chromatids at the metaphase stage of mitosis. And we talk about the P arm, which is a short arm, and the long arm, and also the centromere, which is a constriction in the middle of well, not necessarily in the middle, but within the chromosome, plus also the telomeres. When it comes to the basics, there are several things that you need to be aware of. So the punctuation is important. Just like you need full stops in a sentence, so you need full stops in ISCN. So the full stops separate techniques. Commas separate the chromosome number, the sex chromosome constitution, or the chromosome abnormalities, probes, or they separate the nucleotide numbers to indicate thousands or millions. The semicolon is used where there's a break and join with another chromosome in the short form. The 2 colons are used in the detailed form of ISCN where there's a break and join within or with another chromosome. Plus is used for a gain and minus is used for a loss. Underscore is used for a loss or gain, which is encompassed between the nucleotides, and that's used in array. And the question mark is used where there's uncertain identification of the chromosome regional or actual chromosome. It is not used in array, and it's not usually used in FISH. Tilde is used for intervals or boundaries or for a range, and the / is used to separate clones. We use 2 / or a double slant to separate recipient and donors when you've got a bone marrow transplant and you've got 2 different cell lines. So the terms for acquired are slightly different to constitutional. So when you have an acquired disease, you may also find you have a constitutional abnormality, and then you can use this little small c. You may find that your metaphases some of them are broken, and you're not able to produce an a full karyotype, and so you might produce a composite karyotype, and I'll talk more about that later. And you use the term CP if you've done that. We now know there's, that sometimes what happens is we have chromothripsis, particularly in neoplasia, and the term CTH is used. IDEM is a lovely term which means the same, and it denotes the stem line karyotype in a subclone, and it does make writing the ISCN much easier. Sometimes we have incomplete karyotypes, and then we can use INC. Occasionally you come across homogeneously staining regions, and sometimes they are so complex that we have to use the term CX. Double minutes we use the phrase DMIN, and for a diminished signal we used DIM. Marker chromosomes are referred to as MAR, stem lines by SL, and sidelines by SDL. There's loads and loads of terms, but if you go into pages 34 and 36 in the book, or you go online, you can see all the different terms that you might want to use. I've just chosen some of them. So let's look at some of the basic principles. Normal results, they are far easier to write than an abnormal result in acquired diseases, unless it's a very simple one. So we expect 46 chromosomes, and we've got 23 pairs. 22 are autosomes, which is chromosomes 1 to 22, and we have 2 sex chromosomes, XX or XY, depending if you're female or you're male. And then the ISCN itself is a way of actually writing what the normality is or what the abnormality is, and it can be done for whole genome and for region-specific abnormalities. So for normal results, we give the chromosome number first, and then the sex chromosomes are listed after that. So straightforward for a G-banded karyotype, it's 46XX or 46XY. But for acquired disorders, we put the cell numbers within square brackets of how many metaphases we counted. For an array, you don't have cell numbers, but you would write exactly the same as constitutional, and you'd put the sex chromosome followed by the autosome times 2, and if it's male, obviously X, Y, and times 1, because you've only got one copy of each. For metaphase fish, you write ish for metaphase fish. For interphase fish, you'd write nuke-ish. But for metaphase fish, you'd put ish and then the chromosome region that you're looking at with the probe, and then in brackets, following that, the probe times 2, and then the number of cells you've looked at. If you've got more than one probe, then within the brackets, you'd put probe A, comma, probe B, and then outside the brackets times 2. That's for normal results, and of course the cell number. For interphase fish, the probe is given again times 2 and numbers, and exactly the same for the other normal result. You've got multiple probes. For RSA, again, it's the RSA plus the regions test times 2. So you can see there is some sort of similarity between the different techniques and how you write the normal results. It doesn't always work for the abnormal, they're slightly different, but there are some basic principles. First of all, if you've got G-banded chromosomes, you give the total number of chromosomes, then you'd give the sex chromosome constitution, and then you list the aberrations in numerical order, and you use plus and minus to indicate gain or loss, and you use the abbreviations for the type of structural aberration that you have. Now, when it comes to numerical changes, it's fairly straightforward. You give the chromosome number, X and Y, and then the autosomes in numerical order. So for a case here, you've got 48 chromosomes, there's your sex chromosomes, and then 8 goes before 22, and that's the number of cells you've analysed. When you have multiple clones or sidelines, the 46XX or XY, the normal cells, are always listed last. Doesn't matter if they're the largest cell line, it's different to constitutional, they are always listed last. So if you had an inversion 3, for instance, and then you had an extra X and it was acquired, because you know that because you've got a normal karyotype here, then it becomes plus X. It is not X, XY. It is an acquired change, so it's plus X. However, if it was proven to be XXY, so you'd see that in the normal cells here, that this is the normal constitutional karyotype, and so you put C, and you have to put it here as well, it's XXY, because that's how we write the constitutional ISCN, but it's different if it's acquired, it's plus X. So when we come to structural abnormalities, we get the numerical changes before the structural change in chromosomal order. So in this particular case, you've got an extra X, you've got an extra 16, sorry. And that goes for the inversion 16 because numerical go before structural, and then you've got a plus 22, and you've got a comma between each of the abnormalities and the sum numbers at the end. When we describe abnormalities, we describe them from Peter to Cuta. So I've just put some examples here whereby they haven't got a normal Y. So you have a comma after the X, then you put the structural aberration of the Y from P to Q, and then comma, and then you've got another structural abnormality here. Cell numbers are given for acquired karyotypes and also for the normal results. And so I've just given two examples here where things are optional. So we have this karyotype here. This is the longest way that you could write it. So you've got a 611 recurrent rearrangement in 60 cells. You've got three cells that are minus Y, but still the recurrent rearrangement. You do not have to repeat the breakpoints. It's optional wherever you do. It's easier if you don't. It's clearer to see. And then you've got one normal cell. However, because you've already got this abnormality here, you could just put idem. So you don't put, you just put, if you use idem, you just put what additional abnormalities there are. It makes it much clearer, 45, comma, idem, comma, minus Y. But both are correct. This is just easier to read. If you have multiple structural aberrations of homologous chromosomes, they are listed in alphabetical order. So again, I've given you another example. Now this is a case where the X is coming after the Y, but the Y is normal, which is why it goes first. And then you put the translocation between X and 13, which happens to be constitutional, comma. Deletion alphabetically is before an inversion. So the deletion goes first of the 7 and the inversion goes second. That's the same chromosome, so it has to go alphabetical. The numerical goes before a structural, so +8, del8. You can use short and detailed form, and you can use both in a karyotype. The short form is far easier to understand, but it doesn't stop you from using the detailed form. So if we had the short form, it would be written like this, and if we had the long form, it would be written like this. Aberrations without centromeres are described last and are not included in the chromosome number, because the chromosome number is talking about how many chromosomes there are with a centromere. Rings of unknown origin are described before markers. So to give an example, there are 49 chromosomes. There's your sex chromosomes, commas again. An ISO 17, comma. You can use this to denote the centromere. Plus a ring, plus 2 markers that are identical, plus 3 minutes. With this particular case, you've got different markers. You've got marker 1 and marker 2, so they're different, but there are 2 copies of marker 1. And instead of putting 2 in front, it goes afterwards. And so you've got plus ring, plus marker 1 times 2, plus marker 2, and then you've got 9 to 50, sorry, double minutes in 10 cells. I've already mentioned about C being used to denote a constitutional abnormality. In this particular case, it is a trisomy 21. If you do not know if it's acquired or constitutional because you haven't had the peripheral blood stimulated, so you're not sure, then you'd put a query C. And in fact, this inversion is a known inversion in the Dutch population. You do not describe polymorphisms in ISCN. If you want to know why and all the reasoning, please go to page 53, but they actually will confuse the reader of the report, who will think that they are structural abnormalities and therefore it means there's an abnormal clinical phenotype. Translocations. As with constitutional, you start with the lowest chromosome first and you use a semicolon between the different chromosomes. If it's a complex rearrangement, which happens probably more in acquired abnormalities than it does in constitutional, you follow the movement of the segments starting with the lowest chromosome. So if you've got a 9-22-10, you follow the segments. So it doesn't go 9-10-22 because that's not following the segments. So 9q34 has gone on to 22q11.2, which has gone on to 10q22, and the 10q22 has gone on to 9q34. And so you're following where those chromosomes have gone, the arms have gone. And again, the normal cells at the end. However, there's other parts of ISCN to help you to denote for more complex rearrangements. For instance, here we have a situation where you've got 6p has gone on to 9p, 9q has gone on to the 22, and the 22 has gone on to the 6p. And this, because you've not underlined these 9s, this is from the same chromosome 9. However, if they were different chromosome 9 homologs, you'd probably write this differently, because you've got a 6-9 which is independent from a 9-22, and you're showing that it's a different rearrangement. IDEM or SL may be used to describe subclones, and these are two examples exactly the same, except for instead of using IDEM, which means the same, we've used SL here. If you use IDEM or SL, you do not include the chromosome or the abnormality. You only include the additional changes. So in this particular case, it's an additional derivative 22. So what is a clone? Well, two or more cells with the same gain or variation is considered to be a clone. The exception is where a single cell can be reported, the same clone was found in a previous sample or a follow-up study of the same patient. For loss, because you sometimes get within metaphases, 3 or more cells to have the same loss. Again, there are exceptions. So if you have two cells with identical losses of one or more chromosomes, when there's also a gain or a structural aberration, then you can report it as clonal. So in this particular case, we have lost the X chromosome. Again, you don't have to repeat the points, or you can put IDEM, but it's into two cells, but it's exactly the same structural rearrangement, so this is pretty acceptable. Another way in which you can report clonal abnormalities or prove they're clonal is if you've confirmed them by a different method, e.g. FISH. Composite karyotypes. So how do you know whether or not you've got a composite karyotype or you've got random loss? Random losses do occur, and it's not mean that they're clonal, and they shouldn't therefore be included. So if we had these 5 metaphases, none of these chromosomes are in any of these cells, and so it's just random losses, and they're not clonal, and you shouldn't include them in the karyotype. In these 4 cells, although they have different cell numbers here, there is consistently a -7 and a +8. The others in blue are random losses. And so you write the karyotype not with 42 to 46, because that is all random losses. Use lowest here, which is the -7 in, even though it hasn't got a +8, as 43, and it goes up to 46. -7 +8, and then you put in brackets EPP4. As I've already mentioned, If you have a clone, you give the number of cells in square brackets after the karyotype, and that applies to abnormal and normal karyotypes. You use a slant line to separate the different clones and subclones, or to separate it from a normal cells. When you have chimerism, which is secondary to the bone marrow transplant, you use a double slant line like this. So in this particular case, the patient had a 922 translocation and the donor was female. And these are the cells from the donor, and there's more of those. In this particular case, all the cells came from the female donor, and in this particular case, all the cells are from the male recipient, and the donor cells are not there. You can use Chi if you wish to. It is totally optional. So what is clonal order? How do you decide which clone to put first? The stem line is always listed first, regardless of size. The side lines are listed next. You write the clones in order of complexity, irrespective of size. When you have two stem lines or multiple side lines present, the use of SL1, SL2, SDL1, SDL2 does reduce parity in the ISCN. And so ISCN the committee does recommend that use IDA in such cases. As I've already mentioned, normal cells are always listed last. In the ISCN, there are some employed levels that are recommended. Exceptions can be made if they're biologically significant or clinically relevant in a specific disease. I've highlighted this in yellow because this is an addendum to the ISCN 2020, which will be published soon. It's also a website already. And we also have in the new ISCN an example, a suggestion on how to count the chromosome aberrations in hematology. And this is to harmonize counting of the abnormalities going forward for the different clinical trials so that prognostic aberrations are counted in the same way within a trial. Fish ISCN, this one of the areas where, from GenQA's perspective, we find most ISCN errors. It's quite understandable because it can get quite complicated, particularly in acquired disorders, because you have so many different types of probes and they all have to be done slightly differently. So some of the basic principles are that you list the probes in chromosome order and then from beta to Q. Control probes are not given in the ISCN if there's a normal signal pattern. Obviously, it's abnormal, you have to report it. And the exception being if you're doing anything for determination or you're doing perhaps checking the transplant. Metaphase fish, both for normal and abnormal, the cell number for each clone are given, but there's no denominator. So you just put, like you do in metaphase ISCN, you just put the cell numbers in square brackets. For interphase, for normal interphase result, they can be reported in one string only if the same number of cells were analysed from different hybridizations, and they're then given in probe order. So in this particular case, although ATM and DP53 are one probe set, and then these are another probe set, because you actually analysed 100 cells, you can put them all together, separated by commas, and then an output time to. If you have an abnormal interphase fish, you do have to follow the chromosome order of the probe sets, and the probe sets given in different brackets, separated by a comma. So in this particular case, 100 out of 200 had one TDP53 signal, and by having the denominator, you know the 100 had a normal signal pattern, and in 150 cells, you had 3 copies of the centromere for chromosome 12, but 50 had a normal signal pattern. abnormal interface, when you've got both normal and abnormal cells, the number of abnormal cells is listed over the total number of cells scored for each abnormal locus, i.e. the denominator. So like we have here, and that's just an example here. For break apart and fusion probes, the use of CEN and CON is dependent on the probe type, whether or not the result is normal or abnormal, and I'll explain that in a bit. So let's just look at some of the principles for FISH ISCN. For normals, you put ×2 for both interphase and metaphase ISCN. Different to metaphase that's same, ×2 for both interphase and metaphase ISCN. The metaphase FISH includes the probe location and it's optional for nuke-ish. For bone marrow transplants, you can include both probes. It's optional, but it can be useful for clarity. And again, Chi is also optional. Now for abnormals, this is where it's different. Metaphase FISH signal patterns are plus or minus, etc. And for the structurally abnormal metaphase, you only describe the derivative chromosomes because you know what you're seeing. You've got metaphases there. For interphase, it is ×3 × 1. And here's an example here. For break apart and fusion probes, you have to describe the number of signals first for each probe and then their relative position using SEP or COM. SEP being separated, COM being combined. The description of the FISH results therefore depends on the type of probe that you're using. Locus-specific probes are relatively straightforward. So you can have nuke-ish, just assume this was a single probe. I know it doesn't usually come separate, and there's no copies, 25 out of 100. That means 75 were normal. You can have this situation where you've got nuke-ish, where you've got 25 that are the same here, with no signals at all, a population of 50 with one signal, and 25 that are normal. And then you might have it, if you do it as the probe set, where you've got two probe sets, ATM and TP53, and then you've got the other probe set, which is 12 and the 13 probes. And they have to be separated by a comma because they're different probe sets. And in this particular case, because ATM and TP53, you've looked at the same number of cells, you can put the ×2 outside the bracket. And although TP53 is on chromosome 17, it's still, because ATM is on chromosome 11, has to go before this probe set. And in this particular probe set, they have to go inside. They can't put ×2 at the end because you've got different signal patterns. So ×3 here, ×2, ×2, and it's 50 out of 100. So not all cells have that signal pattern. With single fusion probes with extra signals, it's slightly different. Nuke-ish, where you describe what you see. So you've got two ETV6 signals, RUNX-X1, you've got 3 signals, and then you've got a joining of the ETV6 and the RUNX-X1 signal. You use the Hugo name, sorry, that's extra signals, but you've still got the joining. You use the Hugo G names if they're adopted by the probe manufacturer. Otherwise, you may have to use the probe manufacturer names. Now, in ISCN, it gives you the following information. The clone name is preferred. Alternatively, you can use the UCSC ensemble locus or the gene name. But it's also at the discretion of the laboratory whether you use probe name, clone name, accession number, gene name, or D number. So you really have a free rein as to which you use. The clone name is preferred, but you can use these others. Now, when it comes to the centromeric probes, the DZ reference is preferred, e.g. DZ, Z1, and most probe manufacturers give you that information. However, there are a couple of websites where, and chromosome centromeric probes, where no DZ reference is given, and then you can use the probe name. CEN is not used in FISH ISCN at all. For dual fusion FISH probes, you want to be looking again at the signal pattern. And again, there's no comma because this is one whole probe. So you have got, if you look at this signal pattern here, you have got 3 green signals, 3 red signals, so ABL1 times BCR times 3. But you've got 2 fusion signals. So it's ABL1 con, because it's a fusion probe, BCR times 2. And it's in 100 out of 150 cells, so 50 are normal. And so the normal cells would have this signal pattern, but you don't have to, you shouldn't put it in the ISCN. It's implied by the 100 out of 150. However, I've made this up, as you can see by that box there. If you had a deletion of one ABL1 from one fusion on the derivative chromosome, then you would have a different ISCN. And here it would be, you'd have to put these inside the bracket because they're not the same numbers. So it'd be ABL1 times 2, BCR times 3, ABL1 con BCR times 1. For the breakfast fish probes, the description of the fish result is different if it's normal or abnormal. So for normal, you've got fish L times 2. That's wrong. Shouldn't be M2 there. It's MLL times 2. For if you want to put it in the log form, it's 5' MLL, 3' MLL times 2, and then you write down that 5' is joined with 3' times 2 because it hasn't separated, because it's a break apart. For the abnormal, it shows the separation of the signals. So for the in this particular case, it goes into the long form. You can show it in the short form. So you've got new QKISH, 2 MLL signals, and then the 5' MLL is separated in the 3' MLL times 1 because it's only had on one of the signal patterns. If, however, you've got loss of 3' MLL signals, you've got no separation of signals, then again you have to talk about the 5' and the 3' separately. So you've got 2 5' MLL signals, which is normal, but you've got one B' signal, and you have got 1 5' joined to 3' because you've lost one of the signals. Now that loss could be due to a couple of different factors. It could be due to a deletion, but it could also be due to a truncation resulting in 5' KM2A, which is the same as MLL. It's the CC Hugo joined to a 3' partner gene with loss of the reciprocal derivative chromosome. So the breaker parts, you don't know what your partner gene is. Fusion probes, you do. If you have tricolor fish probes, it gets even more complicated. So the normal signal pattern would be 2R, 2 green, sorry, 2 red, 2 green, 2 aqua. In this particular case, you've got a signal pattern which is 1 red, green, aqua, 1 red, green, 1 green, aqua, due to a translocation, and com is split. So how do you read this? Well, all the context in the probe are described. ISCN given from P to Q in the context of the rearrangement of MECOM, which is here, is split. So you've got NuKish, GoLim times 2, MECOM, because it's split, be 3, and then MYNM times 2. And you write what you're actually seeing. So GoLim was still connected to MECOM, but you've all got a split in the MECOM gene. So MECOM is separated from MECOM, but it's still joined at the other end to MYNM. Hope that makes sense. What do you do at breakpoints? Well, it's optional whether or not you repeat breakpoints in the ISCN, if it's the same as the G-band ISCN. Now, I have 3 examples. There actually are, I think, 4 or 5 combinations of this, which will all be correct. But some are easier to read than others. And my personal preference would be to use this one because it's the more simplistic one. But you could, for instance, rewrite the breakpoints if you wish. We haven't done this particular example, but what we have done is we repeated what we've seen. We've seen the translocation. But no, because if you've got an additional dir4, that would come before the translocation, which is funny. And then you write the fish, and you describe the dir4, and you describe the translocation, because they've got different fish signal patterns. In this particular case, you can put idem, and it makes it much more simplistic. And again, you can do the fish as above. Or alternatively, you could repeat the breakpoints for the translocation in the fish, but it isn't necessary. So when do you change the breakpoints? Well, you change the breakpoints if fish or array clarify or extend the breakpoints from G banding, and likewise you can change the breakpoints on the fish if array clarifies it further. Let's briefly talk about arrays and RSA ISCN. As I've already mentioned, you can combine short and detailed forms of ISCN. When you have multiple techniques, each technique is separated by a full stop. The G banding put first, followed by metaphase fish, followed by interphase fish, followed by array, followed by RSA, and then sequencing. You can list the results using different techniques on separate lines or tabulate complex results. When it comes to pick up clonal abnormalities, if you can pick them up on the array and the RSA, then you can either do them at proportion 0.6, or you put them in square brackets, or you put 2, 3. Either option is allowed. For abnormal results for RSA, other than aneuploidy, if nucleotide positions are available, they should be included. However, if they're not, you can just give a kit name. And the nucleotide can now be listed commas. Both options are available in the ISN. It comes to regional specific ISN. It's not generally used much for acquired abnormalities, but it may be. And so this is how you would write a normal, again in chromosomal order for an aneuploidy kit, or if you've got a hematology MLPPA kit, you just put the kit and then say that was normal. The abnormalities, you get, again, if it's aneuploidy, you partial gain or partial loss. You give the chromosome the lowest to has of X and Y first. So we've standardized that across all techniques now. So you could write it simply, if you've got trisomy 21 on RSA 21 times 3, or you could put sex chromosomes in 13, 18 times 2, and then 21 times 3. Or if it was female, it'd be done like this. But don't forget, if you've got 3 copies of 18, then the X and 13 are times 2, and then the 21 goes after the 18. So it's slightly different. And again, if you have mosaics, you can quantify like this or like that. Now, we mentioned in an earlier talk in this year about the fusion nomenclature. In a lot of the publications, if you've had, for instance, BCR-able fusion, in some publications it is given as a dash, and in some publications it's given a slash, and some probes are also given a slash when they're not fusion. So there was no consensus about how things should be put in publications, and there were issues with both formats due to other nomenclatures. So the Human Genome Nomenclature Committee, the HGNC, with agreement from ISCN and NHGVS, has now agreed that for publications in the written text, it should be written as BCR-able, able1. This is now being published only last week, and this is the URL to get that publication. The paper gives notation to be used for publications in the text, so that in future, when we refer to a fusion gene, we're expected to write this. At the moment, because it's just been published, if the written text report has either of these two options, we will not be making any comments until next year. All formal results, of course, should be given an ISCN 2020, because that is slightly different. Now, when we talk about whole genome arrays, again, the normal results are very straightforward. Sex chromosomes followed by the autosomes times 2. And for the male, times 1 and times 2. In aneuploidy, you only describe the abnormal chromosomes. You do not describe anything that's normal. So I've given some here for complex acquired array results. And this would be one we would use XY 1 to 22 CX, whereby there are multiple complex rearrangements across the whole genome. If, for instance, it didn't include the sex chromosomes were normal, you take out X and Y, like this. If, as an example, chromosome 22 was also normal, then this would be 1 to 21 complex, or in this case, if X and Y were still involved, but not 22, it would be X, Y, 1 to 21. So you do not mention anything that is normal. And when you've got chromothripsis, again, you just talk about the chromosomes that are involved, and then you put CTH at the end of it. There is no genome build given for normal results or aneuploidy. It's only given if the nomenclature has nucleotides given in it. So when we come to abnormal structural aberrations, then we have got nucleotides, and you do give the human genome build. The short description only includes the abnormal nucleotides. The detail system includes the bordering normal nucleotides, because there's obviously sometimes a gap between these two numbers here. The underscore is to show you the nucleotide range that's involved in the loss in the gain. And the platform information should still be placed in the description of the report. It's not part of ISCN. Now, some of you will be doing optical genome mapping, and the ISCN Standing Committee is well aware of this new technology. There is an ISCN forum, which I'm going to discuss in a minute, which has suggestions for the genetic community to comment on some suggestions for OGM nomenclature, and a working party is to be initiated, so we can come up with something that will work for everybody. But it will probably go on the forum so that we can have more input from everybody else before we actually publish it in the next version of ISCN, which won't be for a few years yet. So if you've had chance, you may have gone on to the ISCN online via the Cargo website. Now, to access it is a very small little icon at the bottom here which says Access ISCN Online. We have spoken to the Cargo and asked them to make this a bit more clear so that you know where to go. And when you click on that link, you'll get this sort of page where you've got some information up here, and you've got it also here. So if you clicked on this, the table of contents, then you will get all the information about the different chapters, and you just scroll down to see the different chapters and the content. You will also notice there's something here called the forum. And the forum is where you can put questions, start a new question, or click and see an old answer. And hopefully this will help us as a committee to develop and improve the ISCN over the years, and also give answers where you've got questions. So this is one where there was a question about the interface fish for gonadal status using sequencing. Oh no, it wasn't, it was another one, sorry. Unfortunately, when it was published, ISCN 2020, we found some mistakes. So some of you have been very kind and letting us know what some of these mistakes are. So now when you scroll through the chapters, you will be able to see where if there has been a change on in the ISCN. Now, the errata will also be published as a whole in Cytogenetic Research Journal very shortly. Now to look at before it goes to press, and then you can have a list of everything all together. You mentioned, if you heard before when we've given these talks, that there's going to be an ISCN pilot EQA in 2021. We are using a new online interface, totally different look, and is hopefully much more useful for you and easier for you to navigate. as a consequence of this online interface and some of the developments that need to be done to enhance it, we've had to delay opening until November 15. But when it does become available, there'll be constitutional cases, both with karyotype ideograms and fish images. Specialized laboratories may participate in a subset of questions. You can submit your ISCN answers online. You'll have 5 weeks to submit your results. There'll be probably 4 questions for constitutional, 4 for acquired, 4 for fish. And the assessment panel will include ISCN experts. It's a pilot, there's no performance monitoring. And at the moment, you can still enroll. So we now come to quiz. My first question is, which of these is correct? Now, you're going to have to remember whether it's A, B, C, or D, because when you get the link to actually submit your answers, you won't see the question. So if you look at that quickly, make a decision, and my colleague Melody will put up the option for you to select A, B, C, or D.
Yes, thank you, Ros. So we'll just give them a quick minute to select the right answer, and then I will launch the poll, and you'll be able to select A, B, C, or D, whichever one you think is
correct. I think they can do it now.
Right, okay, let's launch it then. So you should be able to see the poll, and you can make your selection. Okay, so we'll close that now. I can see that quite a lot of people have voted. And I can share the results. So we can see that 2/3 of people listening have voted for answer C. Okay, so I'll just hide that screen. It should go back to the PowerPoint, and Ross can explain the correct answer.
Can you see the PowerPoint?
It's not on slideshow, I don't think, anymore.
Okay, let's do slideshow.
Resume slideshow at the top.
Got it.
Yeah.
So, hopefully you've all selected C. Because
so we had 2/3. 2/3 selected C.
Okay, A is wrong because you haven't given the cell numbers. This is an acquired karyotype. B is wrong because this actually is not a plus X, because you've only got 2 X's, so therefore it should be XX, and it should be 48, not 49. If it's 49, it should be 49XX plus X. C is correct, and D has got too many spaces, and we shouldn't really have spaces, although I know it's not always easy using some of our lab computer systems. Now we have a total of 7 questions, so we'll go now on to question 2. So which is the correct ISCN order? You've got 10 metaphases, you've got a ring chromosome, you've got translocation involving 821, you've got +8, you've got +X, you've got -21, you've got a del1, and you've got 2 different marker chromosomes. So look at A, B, C, or D.
Okay, I'm going to launch the poll now and select your answer from A, B, C, or D. Okay, a few more seconds, as I see about 60% of people listening have voted. Okay, I'm going to close this one and we'll see the responses. So we had 2/3 people selecting answer A, but also 20% selecting answer B.
And it is answer, it's gone back.
Yeah, it keeps going back to the...
It is answer A. You said some said it was...
We had 20% saying B.
Right. It's not two markers because there were different markers. And ring goes before marker. So that's why that one is incorrect. This one's incorrect because it's plus X is in the wrong place. And this is incorrect because you've got numerical before structural, and that doesn't work. So it's just to remind yourselves that it's sex chromosomes first, autosomes in numerical order, numerical before structural, but remember it's autosomes in numerical order. So if it's the same chromosome, then numerical go before structural. It doesn't mean that all numerical go before structural for every chromosome. And multiple structural changes, alphabetical order, so inversions go before translocations, derivatives when then centromeres would then go next, and unidentified abnormalities, rings, markers, and double minutes. Aberrations within the same chromosome, again, from Peter to Cuta. So let's move to the next one. This is quite a quick one. So which is the correct karyotype here for a deletion of 5q?
Okay, yeah, so we just had a comment asking for a little bit more time to see the questions. So I'll just give you a bit longer to think about this one. I guess the last one was a bit more complicated as well.
It was a lot more complicated.
Okay, so I'm going to launch the poll now. You select your answer. Okay, so we'll close this one. And we've got the vast majority selecting option B, 76%, but also a few selecting on option C.
Right, option B is correct, and option C is not correct because you don't put a semicolon in for deletions, duplications, or inversions within a chromosome in the short form. You do in the long form, but you don't. In fact, use colon there, but you don't do it in the short form. And this particular one, we don't use del 5q. It's only used in the text. That's the colon, and then this, obviously, the normal cell line should not go first. It should always go last. Now, this is slightly different. For abnormal closed stem line, always goes first. For constitutional, the cell line's the largest abnormal cell goes first, which I think is why if you're doing both acquired cytogenetics and constitutional, there's sometimes a confusion about what the order of the cell lines or clones are. But with acquired abnormalities, it's always the stem line first, regardless of size, followed by the next sideline, and increasing complexity as you go through the different clones. So let's do the next question. Which is the correct karyotype? This is a three-way translocation where 9q34 has moved to 22q11.2, 22q11.2 has moved to 6p21, and 6p21 has moved to 9q34. There's an additional DIR to 22, and there is one normal chromosome 6, one normal chromosome 9, and one normal chromosome 22. A, B, C, or D?
Okay, so think about the order, the number of chromosomes and the punctuation, and select your answer on the poll. Oh, sorry, wrong one. That was the last one. Sorry about that. So you can vote now, thank you. This one might be a bit more tricky. Okay, so I'm going to close this one. Show you the results. So we had just under half selecting option A, about a third selecting answer B, and a few selecting C and D. So quite a spread on this one. That was question 4.
Well, when there's more than 2 chromosomes involved, you have to start with the lowest number. So you have to start with chromosome 6. And the older the chromosomes is then dependent on the movement of the chromosomal segments. This one is the 9 and 22 are both wrong because it starts with the lowest number first, and this one is wrong because you've got an additional 22 and should therefore have 47 chromosomes. We've only given you 3 options here because we're on fish ISCN and it takes a lot longer to think about. So basically, you have got loss of TP53 and you have got gain of chromosome 12. So which of these is the correct ISCN, A, B, or C?
Okay, I'll give you a bit more time to think about this one. There's a lot to think about with the fish ISCN, as I also just said. Okay, so hopefully you've decided between A, B, and C. And you can vote now. Okay. So we've got 73% selecting answer C, and the remainder split between A or B.
And the answer, oops, try again. And the answer is C. A is incorrect because you need to put the probe sets together. So ATM and TP53 should go together. This has just been done in chromosomal order, which is incorrect. You have to put the probe set together. And in this particular case, this is incorrect because you've got it totally out of order. ATM doesn't belong with this probe set and TP53 isn't separate from this set. So this is correct because you've got this one probe set, this is the other probe set, and you've got the right number of cells for each one. So you can keep the probes that are sets are co-hybridized in the same parenthesis. Because they are different probe sets, you have a comma, and the probes are given according to the chromosome number. So 12 comes before 13, and in this case 11 comes before 17. And this always doesn't seem to make logical sense because 17 obviously should come after these, but it's talking about this probe set first of all. So which is the correct ISCN for this dual color dual fusion probe using BCR and ABLE, you can see the signal pattern here. You've got one normal ABL1, one normal BCR, and 2 fusion signals. So it's A, B, or C.
So again, check the number of signals, the punctuation, decide between A, B, and C, and you can vote now. Okay, so I'll close this one. And we've got most people selecting answer A, but quite a few with B or C as their thoughts for the correct answer.
A is correct. Well done, folks. B is incorrect because you actually have 3 green and 3 red signals, so it should not be ×2. And this is incorrect because there's a comma here, which it shouldn't be because it's one probe set. So the next one, triple color probes. You remember what I showed you? I need to choose from here, A or B. Remember that you've got a 1 red green aqua, 1 red green, 1 green aqua, and you've got mecon that is split.
Okay, so this is our last question. Decide between A and B, and vote now. Not so many voting on this one, but triple colour probes are not used as often.
And I think people are getting used to the nomenclature for it.
So we've got 2/3 going for option A and about 1/3 going for option B.
Well, option A is the correct option. The reason this is incorrect is because you actually have 3 mecon signals, so you can't have ×2 there. And this is the agreed nomenclature that was agreed by the ISCN Standard Committee. You don't put ×2 here, ×3 here, ×2 here. You have to actually explain what's happening with the different signals, not just the mecon signal, because you need to know what it is, how it's working in relation to the other 2 contigs. So you're describing the ISCN from P to Q in context of the rearrangement. I hope that makes sense. So just to say thank you to the ISCN Standing Committee, Cargo, who have done the online version of the ISCN, obviously published the booklet, GenQA staff who have all given examples and have helped in the preparation of this talk, plus yourselves, because without you, we would not be improving the ISCN, and we do need your constant feedback. So thank you very much for all your help. And I think I'm now passing over to Melody, am I not?
Yes, so we're running a bit short of time. We've had a few questions submitted to the website. We'll try and address a couple of these quickly. If we don't get round to all your questions, we'll either reply individually or publish something on the website. So one of the quick questions is, somebody saying, thank you for the webinar, it's very, very useful. What's the difference between IDEM and SL use?
There isn't any difference. You can use either. IDEM just becomes is easier to use. It's also, when you've got a very complex carrier type, it is the preferred option by the standing committee, but you can use either.
Thank you. And this is an interesting one. Somebody asking if there is a moment or in the future any special ISCN guidelines for mesenchymal stem cell carrier typing.
Ooh.
So I guess to distinguish that they're stem cells.
I would have thought you'd have just used the normal G band, assuming you're G banding or you're doing array, whatever you're doing, that you just use the same nomenclature. Because if you think about it, ISCN is being used for, doesn't matter what the tissue is, you're using it for bone marrow, you're using it for peripheral blood, you're using it for amniotic fluid, you're using it for CVS, so stem cells are the same. I would have thought we needed to have it separate. But if you think I've got that wrong, please give me the reasoning why, and we'll look at it.
Okay, let's just see how many more we can get through. So this is an autograft scenario. So all the cells from the same patient, and if they're all normal after the transplant, do you still need to put the 2 slashes, so 46XY with the slashes to show that it's a transplant?
Yes.
Okay, that's a simple one.
Sorry, very simple answer.
Yeah, I'm just having a look, and there's a few more just come in on the last minute. I think we might have to address these ones individually. Sorry, I can't see them all on the same page. Could you please explain a little bit more on the question with the DIR22 and the correct answer, please?
Which question is that one? Let me have a look quickly. Question 4. Okay, so the lowest chromosome number has to go first, which therefore leaves you only options B and C. And the chromosome number in C is incorrect because you've got to derive 22, and therefore it has to be 47 chromosomes, not 46. So the chromosome number should always be 47. And as I say, the other 2 are incorrect just because you start with the 922 instead of the 6. It's just that the rule is you start with the lowest chromosome first.
Okay, I think we're probably running a bit short of time now.
So let's answer the questions by email then.
Yeah, and there's also the forum as well, which Ros has mentioned on the Cargo website.
Yes, put your questions on the forum. Great place to put them.
Okay, so to round up, thanks very much, Ros, for an excellent presentation, very clear. And just to remind people that if you did miss any of it, or if you wanted to watch it again, the webinar has been recorded and you'll be able to access that to view at your own leisure. Any of your colleagues who you think that this webinar would be useful for, if they haven't registered for it, in about a month we will upload the webinar recording to our YouTube channel, which will be freely available for anybody to view. Well, obviously we always welcome your feedback, and you'll be sent a survey automatically by the webinar platform, and we'd be grateful for any feedback that you have on this webinar, or perhaps any ideas for future webinars that we can help with. The website will also automatically send you a certificate of participation in the next day or so. And our next webinar will be, sorry, I think that one's a bit out of date. Our next webinar will be on prenatal scenarios, and we'll be advertising that in due course. So thank you everybody for attending. Hope to see you at our next webinar.
Thank you. Bye.